Dr. Bar, are you ready?
Sure.
Okay, I just want to make sure that I can do the introduction. So I wanted to welcome everybody to our first CPS core conversation of 2024. I'm very excited to introduce Dr. Katharine Bar from the University of Pennsylvania for tonight's talk, which is titled The Impact of Acute and Early ART Initiation on Autologous Neutralizing Antibody Responses to HIV-1. Dr. Bar did her undergraduate education at Northwestern and her medical school at the University of Iowa Carver College of Medicine. She completed her internal medicine residency at the Medical Colleges of Virginia Hospital, which is part of the VCU system, and her ID fellowship at the University of Alabama. Subsequently, she joined the University of Pennsylvania, where she is currently Associate Professor of Medicine in the Division of Infectious Diseases, as well as as the director of the Penn CFAR Virus and Reservoirs Technology Core. She's a physician scientist with a focus on HIV, and her research relates to basic mechanisms and translational impact of viral transmission, pathogenesis, and persistence in HIV, SARS-CoV-2, and other viral pathogens. Her lab's current work is focused on three broad areas: understanding viral dynamics of HIV persistence and reactivation, developing new broadly neutralizing antibody-based interventions, and then leveraging novel non-human primate models to test prevention and cure interventions. And as we were just speaking about, she's also still a practicing infectious disease specialist and has a primary focus on HIV. So Dr. Bar, thank you very much for graciously giving us some of your time tonight, and we're very excited to hear your talk.
All right, well, thank you so much for the kind introduction. It's a pleasure to be here. I'm going to talk about a project actually that I've been working on with Rebecca Lynch, who I think maybe on here somewhere. Rebecca L. Maybe that's you. Maybe there's another.
That is me, if I'm allowed to say that. Rebecca L says hi.
All right. Hello, Rebecca L. Anyway, Rebecca and I have been working together for a few years now. And honestly, it is one of the things that makes me wake up and go to work in the morning, because she's super fun and smart, and I appreciate working with her very much. So anytime the DC CFAR asks me for anything, I'm dedicated my psychological health. I owe a lot to you guys for letting me steal some of Rebecca's time. Okay, so I'm going to try to share my slides now. How about that? All right. Does this show up?
Yep.
All right.
Very nicely.
I'm going to do my laser pointer. Okay. Here we go. Feel free to interrupt me, ask questions whenever you want. Tell me I'm wrong, whatever. Keep it lively. I'm sure everyone's tired or hungry, and try to make this interactive. All right, so as you are all aware, antibodies are central to effective immunity across viral pathogens. We know that in our vaccines, for example, antibodies are the correlate or co-correlate for protection in almost every licensed vaccine that we have to date. Similarly, even in really complex, diverse RNA viruses, we know that antibodies are now a correlate of spontaneous clearance. So this is data from a hepatitis C study showing that more and more we understand not just cellular immunity, but the development of effective antibody responses is associated with virus clearance. And this is against hepatitis C that is very diverse and has a lot of the sort of challenges that HIV has, and yet we know antibodies are relevant. But HIV is absolutely not that case. HIV is perhaps the most highly evolved viral pathogen against neutralization, and there's a bunch of reasons why. But for the most part, HIV's the antibody response in HIV is ineffective or too little too late at every standpoint. So if we think about virus neutralization as binding of an antibody to the functional trimer, preventing it from infecting the next cell, then there's a bunch of reasons why HIV is highly evolved to evade neutralization. So this is a schematic of a virion, which is a nice cartoon and an advertisement for Croy, which I hope everybody comes to this year. But it actually represents a lot of what is unique about HIV as an enveloped virus. So on the virion surface, in addition to having a small number of functional trimers, there's actually a whole bunch of non-functional species that are decoy immunogens. So there's GP41 stumps, there's GP120 monomers or dimers, There's OMV trimers that are sprung open and are not in the right energy state or in the right conformation to confer infection. And all of those things are highly immunogenic to the immune system. And so our early antibody responses are much more interested in those things screaming, I'm a virus, than the glycosylated trimer, which is it's the most densely OMV protein that we know of in our enveloped viruses. And its broad and conserved epitopes are actually tucked really down in recessed cavities, and so they're very difficult for the immune system to see. We also know that HIV kills CD4 T cells, so it kills them directly by infecting and killing them, but there's also a ton of bystander killing of CD4 T cells. And so that CD4 T cell help is essential for antibody maturation, and we lose that in HIV specifically. And then finally, HIV is incredibly diverse. So the diversity of the virus within any one individual or any one region, or certainly the whole global pandemic, is very hard for any one antibody to be able to see and be effective against. And so for all of those reasons, HIV's antibody response is too little, too late. So if we look at the well-described kinetics of antibody responses, we know that our first antibody responses are cross-reactive antibodies, again, to one of those decoy immunogens, GP41 stumps. So these are antibodies that have likely developed to gut microbes, but have some cross-reactivity. And so we see very early responses first against GP41. And then we see binding antibody responses against GP120. So some of those non-trimeric OMV species. And again, these things are very important for diagnosis and for us to understand when people are recently infected, but they do not prevent ongoing infection. They do not determine disease prognostication. They do not neutralize the virus or clear the virus. So we see these non-neutralizing GP120 antibodies start to develop in the first sort of 2 to 4 weeks. And that's when we can start to diagnose HIV infection. But we do not see our first neutralizing antibodies until 3+ months of infection. And that is way too long of a window to have those antibodies affect cure or clearance or even virus control. In addition, by the time those neutralizing antibodies arise, the virus has already escaped. They target strain-specific, highly mutable regions on that envelope, and the virus quickly reacts to that slow-going immune response and escapes. So by the time you detect even low-titer neutralizing antibodies, the virus that antibody is targeting is long since escaped. So we have this slow antibody response and fast viral escape that goes on and on and on and on for however long this individual is viremic and not on antiretrovirals. And we know in a small subset, somewhere between 10 and 30% of individuals, that back and forth antibody and virus escape pattern leads to the development of these broad or elite cross-reactive neutralizing activity. Now, by the time that's happened, the virus in those individuals has almost certainly escaped, and those BNAbs tend to be ineffective in the individual. But those are the kind of antibodies that we've pulled out and used as our prototypic BNAb monoclonals, and we use these in a whole bunch of ways for both our targets of vaccination and then as therapeutics in and of themselves. But the theory of using antibodies in HIV is to change that natural relationship and take those elite cross-reacting or best BNAbs that have broad and potent neutralization and good effector capacity and give them at a different clinical juncture before the virus has time to escape. So if we think about vaccination, that means to deliver BNAbs or vaccine-induce the immune response to make these broad antibodies so that they're there before any individual is challenged with the virus. When we think about BNAbs for treatment or cure, if we're talking about vaccine, that means taking that baseline autologous antibody response, giving antiretrovirals that block that virus from escaping, and then as the virus is locked down, asking the antibody response to further mature. So we have good, broad, potent specificities, and our virus hasn't been able to escape to adapt them. So those are the theoretical ways we use antibodies as in HIV. Now let's switch just a minute to early antiretroviral therapy. So we're all aware that standard guidelines are for any person with HIV, we should start antiretrovirals as soon as possible, regardless of clinical disease, regardless of their the state of their infection. And that's because we know that it's a huge benefit for that person's health. It stops disease progression, it returns them to health. It's essential for that individual's health. In addition, there are public health responses or public health rationale for treating individuals with HIV. We know that if the virus is undetectable, U equals U, undetectable equals untransmittable, and we can limit the spread of ongoing transmission through antiretroviral therapy. We also think that early antiretroviral therapy has benefits that may create this optimal setting for an immunotherapeutic cure strategy. Should we develop one, that would be a great time to use it. And that's because we know early antiretrovirals restrict the size and the diversity of the latent reservoir. We know that early antiretrovirals protect humoral and cellular immune responses. It prevents the development of exhaustion and dysfunctional immunity that happens with chronic infection. We also know that people who start antiretrovirals early are much more likely to go on to have post-treatment control than those who start antiretrovirals later. So all of these reasons think this may be a great setting for cure strategies. However, we don't actually know how these antiretrovirals are going to affect autologous antibody responses. So just one more thing about ANAB, autologous neutralizing antibody activity and post-treatment control. So there are rare examples of autologous neutralizing antibodies being the correlate of a person who goes on to have spontaneous control of their virus after treatment interruption. So this is a report from Taeuk Chun and his group at the NIH. And they characterized multiple post-treatment controllers. And this person, participant 30, what they found here was that this is starting from the time of ART initiation. And again, we're sampling the virus in their triangles, and there's very few low-level virus detectability, but mostly suppressed for many, many years off antiretrovirals. And what they did was they took plasma antibodies and asked, does this neutralize the reservoir virus or these small little blips of rebound virus? And what they found was the autologous antibodies were very potent against all of the reservoir viruses they could find until this point right here. And what in fact happened was this individual experienced super infection where they had a different variant, very distinct from what their antibodies could target, that was transmitted, and that was enough to break through this the antibody response. But this is a good example of autologous neutralizing antibodies being a correlate of spontaneous clearance. So again, we don't know how early or acute ART initiation affects the development of neutralizing antibodies. And so that's sort of the question we wanted to ask. Now, this has been asked a little bit. So the Military Health Research Program, the MHRP, has these amazing cohorts of acute and early ART initiators throughout the world. And they've looked at some of these questions. So particularly, they looked at binding antibodies and ADCC. So this is cohorts of individuals who were started on antiretrovirals during stage 1, stage 2, stage 3, or stage 4 or 5 of disease. And that largely means, did they just have virus present and no antibody responses? Did they have very early antibody responses, or did they have slightly more mature antibody responses? And here, looking at the titers of GP120 specific antibodies, they showed that basically once you start to develop early antibody responses, these can grow a little bit over their week 0, 24, 48, the first year of antiretrovirals. So they do see binding antibody responses in early ART initiation. Similarly, they looked at ADCC, so the ability of these antibodies to recognize and kill virus-infected cells. And these data are a little messy, but basically at later early infection, the antibodies' presence in these individuals seem to have some ADCC effect. Then they looked at tier 1 neutralization. So this means they took basically lab-adapted viruses, MN, which is a clade B, and this virus, which is a clade E envelope. These are not primary viruses. These have been changed in the lab so that they're very easy to neutralize, but they're kind of a signal for any hint of autologous neutralization. And they were not able to detect, this is all baseline reactivity, so they were not able to detect any neutralization in any of their early ART initiators. But they did not look at autologous virus. They did not ask, are these antibodies able to neutralize their own person's virus? And so we decided to try to ask this question by partnering with Dr. Sugi Lee from the UCSF. And so the UCSF has this treat acute HIV study in which people coming to UCSF or the public health department are identified to be an early or acute infection, started same day antiretrovirals, enrolled into this clinical cohort where they're followed over time and they have samples collected as well. And so from this group, we had 23 participants and we divided them into 3 groups a little bit randomly. But the first group were people who had an estimated time of infection before diagnosis and ART of 60 days or less. The second group had 60 days to about 120 days. So this is sort of our acute ART initiators or early ART initiators. And we grouped them like that because we thought they were likely to either not have any neutralizing antibodies or likely to have neutralizing antibodies. But again, it was just a guess to begin with. We also have a third group of individuals. And so we're following sort of this gray, red, and blue throughout the rest of the talk to denote these groups. But in the middle, in red, these are acute ART initiators who, after starting on antiretrovirals, had some detectable viremia. So we have one individual with an early blip at about, I don't know what this is, 15 or 16 weeks, and then 2 individuals at the end here who had detectable virus at the end. We know now from talking to the providers that likely this was associated with non-adherence to their medications. We have drug levels to sort of back that up. But early on, we weren't sure why they had viral rebound. We just knew that they had detectable viremia, whereas the rest of our cohort had no new sort of detectable viremia. So the thing I want to point out is that the kinetics of viral decline on antiretrovirals is different in acute and early ART initiators. And this is well described because we think of the sort of logarithmic decline of virus. And our acute ART initiators are generally starting much, much higher. So we have viral loads of 1 million to 10 million copies per mil in many of these individuals. And so we see virus decline over time. In many of these folks, it takes 6 or more months before our virus is undetectable by our clinical assay. And that has been seen in a lot of acute ART cohorts. We know the virus is very high. There's a lot of virus-infected cells. Some of those cells take longer to decline. As opposed to our early ART initiators here, down here in blue, where we have lower viral loads in general, we sort of reach closer to a set point viremia, and our several log decline in viremia takes us under that undetectable level a little bit faster. But the reason I point this out is because even in our acute ART initiators who are very adherent to their antiretrovirals, there's no ongoing virus replication, but there is still virus in the system. There is still virus detectable in the plasma and likely in the tissues for many, many months, at least 3 months, if not 6 months or longer in many of these individuals. So I think that's an important thing to consider. So we started by looking at binding antibodies. So on the x-axis here is each one of these 23 participants' first sample. So the day that they were enrolled into the cohort and the estimated time from infection to starting antiretrovirals. And so this is each person's baseline sample and saying, do we see evidence of binding antibodies? So we're looking at GP120 binding. We used a YU2 envelope, but this is sort of a semi-quantitative assay. And you can see we start to see binding antibodies at about 30 days. And then that kind of increases over the first 3 months of infection until we plateau at about 90 days or afterwards. If we look at how that evolves over the time of follow-up we have in these individuals, each one of these individuals has between 6 months and several years of follow-up on antiretrovirals. For our acute ART initiators, there's not really a consistent pattern of how these antibodies change. Some go up, some go down. Similarly, for our early ART initiators in blue, most of these antibody levels stay stable, a few decline over time, but we see a substantial increase in our 3 acute ART initiators who had rebound viremia. So we see baseline lower titers that go much higher after that viremic time point. So then we looked at tier 1 neutralization. So again, these are sort of lab-adapted viruses that are much more sensitive to neutralization than primary isolates. We looked at 2 clade B tier 1 viruses, MN and SF162. And again, what we see with both of these viruses is after about 90 days, we can start to see tier 1 neutralization. Again, in our acute ART initiators, none of these individuals had tier 1 neuts at baseline or throughout their entire time on antiretrovirals. Those who had viral rebound, though, did develop these tier 1 responses. And in our early ART initiators, we see most of the individuals have tier 1 neuts and they were relatively stable over time. So in order to look at the autologous neutralizing response, we have to characterize the autologous virus. So we took plasma from that first day and we sequenced the envelopes, but using single genome sequencing. And then what I'm showing here is the phylogenies, the phylogenetic trees of the GP160 sequences from 2 representative acute ART initiators. So here, what we can see in this phylogenetic tree is that these are basically identical envelopes. These down here have a very small amount of diversity, but this is what it looks like when a single virus is transmitted, establishes infection, and over a small period of time has a little bit of random diversification. But basically, we can isolate from this information, we can say this identical virus population, this is the sequence of the virus that was transmitted and founded infection. This is the transmitted founder virus. And we can clone that virus in yellow and use that to test for neutralization sensitivity moving forward. Here we have a similarly acute ART initiator, and yet when we look at the phylogeny, we see 3 low diversity lineages. And what this represents is the transmission of 3 distinct viruses that have had very little virus replication such that they are not recombining at all. And we can identify clearly this is 3 transmitted founder viruses. We can clone 1 virus from each of these lineages and move and use that in testing. Now, when we sample those early ART initiators, it becomes a little bit more muddy. So this is a single virus transmission sampled at about 90 days. And so what you see is that it's still not a ton of diversity, but there's a bunch of these lines here that represent shared mutations. So some of these sequences have mutations that are shared, that are distinct from each other. And what that represents is selection. So there's some reason why these viruses are choosing. There's an advantage to the mutations that are part of these of this diversified tree. So we cannot infer back to the exact transmitted founder virus here. And so we sort of sample from known lineages and try to say these are representative OMB sequences. We also know early diversification in OMB like this that is non-random. There's shared mutations here. We can infer that that's from selection to immune pressure. And in OMB, the #1 immune pressure is autologous, is neutralizing antibodies. And so we can guess, but we can't be sure, that this degree of virus evolution suggests the presence of neutralizing antibodies and that this virus has escaped from it. We don't know for sure, but that's what we would guess. Here is another participant with multiple virus transmission sampled in that early period. And what you can see is a huge amount of diversity. And this is basically what happens with multiple virus transmission and with enough time for cells to be multiply infected, get a lot of recombination, and all of a sudden you have diversity that's very similar to a chronic ART a chronically infected individual. We have lots and lots of diversity. It's difficult to infer back on exactly which virus is from, which virus started this, which is a recombinant, etc. And so we just sampled from 2 sort of areas of the tree in order to try to sample these early viruses. And honestly, we made a lot more clones than this, but many of these viruses were non-infectious. And so we're just using the ones that we were able to make, infectious pseudovirus from. Okay, so this is for the phylogenetic modelers in the room, I guess. I think this is very cool, but it may not be that exciting for everyone. So basically what we wanted to do was use our phylogenetic trees and model the time that from our phylogenetic modeling using most recent common ancestor assessments to say, based on the diversity in the trees, how long do we think these individuals have had replicating virus? And we can compare that to the clinical estimates. So the clinical estimates are based on, have they had any negative tests in the past? When do they think an event might have happened? What does their antibody testing look like? Do they have a 4th generation positivity, etc.? So all of our clinical information provides an estimate, and that's shown as a black X. And our phylogenetic tree provides an estimate, and that's shown as an orange circle. And so for our acute ART initiators with single transmitted founder viruses, you can see these align beautifully. They're very, very similar, and that makes us think our modeling is correct in both regards. With our multiple virus transmissions, again, if we look at the diversity from the whole phylogeny, it's a way overestimate. But if we just look at diversity in green triangles of one of those lineages, then those match very nicely. Again, when we look at early ART initiation, it's a little bit, it's still pretty good for our single virus transmissions, where our clinical and our phylogenetic estimates are nicely overlapping. And our multiple virus transmissions, it's just too much. We can't really pick out a single clade to be able to do these analysis, so it's not that helpful. The purple stars here are looking at a model of virus evolution that suggests if basically autologous neutralizing antibody escape has happened. And so if we have star-like phylogeny, that basically means only random mutations have arisen and no selective pressure is forcing the virus to choose one pathway. And so if we look at the single lineages of our multiple viruses or our single TFs in the acute ART initiation, for the vast majority of these participants, we do not see evidence of virus escape from an adaptive immune pressure. And this suggests we don't have sequence evidence of antibodies, neutralizing antibodies. Whereas in our early ART initiators, we see selection in this virus population, and we can guess that that's autologous neutralizing antibodies. We don't have definitive proof at this point, but we do not have any star-like phylogeny or only random virus evolution. Okay, so now we can take those transmitted founder viruses from our acute ART initiators and our early OMS likely escape variants, and we can test for neutralization with our plasma collected over time. So at our baseline enrollment time, we're basically asking, does contemporaneous plasma antibody neutralize the virus replicating at that time? And we expect there to be none, because either those antibodies haven't developed and therefore there's nothing to neutralize the virus, or the antibodies have developed, but the virus has escaped. And therefore, in all of our individuals, we see no detectable neutralization from the plasma at the time of enrollment. But what we're asking is, on antiretroviral therapy, do we see that response continue to mature as we've stopped virus replication? And so at week 12, we don't see any activity in our acute ART initiators, but in our early ART initiators, in 4 out of the 8, we start to see some neutralization of this original virus by this later plasma. By week 24, we have even more reactivity. And by our final time point, 7 of 8 of our early ART initiators have expanded their neutralizing antibody on ART to target that original virus. None of our acute ART initiators have any neutralization. And 2 of the 3 individuals that had viral rebound have started to develop neutralizing antibodies. So again, none of the participants with acute ART initiation who maintain suppression developed neutralization. And we think about the fact that some of those folks had virus on board for 6+ months, and yet they did not develop neutralizing antibody responses. But 2 of our 3 individuals with rebound did. So here's our one individual who had rebound at about 15 weeks. The next sampling time point, we have low-level neutralizing activity. This individual, at the same time point we detected viral rebound, also had neutralizing antibody activity. And this person down at the bottom had a rebound but did not develop neutralizing. So even though the acute ART initiators who maintain suppression, none of them develop neutralizing antibodies, but a little bit of viremia is enough to push this cohort to developing. So we suggest that this is like a primed response that is ready with just a little extra virus replication to be pushed over into making neutralizing antibodies. All right, so let's look at the early ART initiators. So this is our baseline time point. Over time, we have the development of increasing potency against the early ART virus over time. And so there's a variable amount of follow-up for these individuals, but what we can see is over years of antiretroviral therapy, the general trend is for these responses not only to develop, but to increase in potency years after they've been suppressed, or months to years after they've been suppressed on antiretrovirals. So this suggests that antibody response continues to evolve and mature over time. So this is the coolest part, in my opinion. In the early ART initiators who have multiple virus transmission, we can not only ask about the potency of that response, but we can ask about the relative breadth, because they actually had a fairly diverse virus population at the time they were initially sampled. So this individual, 8012, has a lot of diversity at the time they're first sampled. Here we're looking at the bottom, this gold virus, and at the top, this green virus. This is a highlighter plot of the amino acid differences between these sequences. And what you can see, I hope, is that there's a lot of difference between this green virus and this orange virus. In V1 and V2, we see diversity. All throughout GP120, there are multiple amino acids that are different. So an antibody that can detect one of these viruses may have trouble finding that same epitope in this other virus. Okay, regardless, what we see again with our antibody response, if we look at these two viruses, is that at baseline, we don't have any response. By 12 weeks, we start to see the neutralization of this gold virus down at bottom. That increases at 6 months, 24 weeks. It is about the same at about a year. Our next sampling time of 80 weeks, however, we see a bump in that potency, and it picks up both of these viruses, the green and the gold. And then those continue to rise slowly over time. So this suggests not only an increase in potency, but an increase in the relative breadth of this response. I personally think it's very interesting for us to further dig into whether this is one antibody lineage that's continuing to mature and expand to be able to better target multiple viruses, or whether this is the evolution of additional BNAB lineage, or sorry, antibody lineages that are now able to target diverse variants over time. The fact that this virus is, these two distinct viruses are kind of targeted with the same potency over time suggests to me that this this is a single sort of reactivity, but I don't know that. And we're going to have to map these responses and the B cell evolution to figure that out. Here's another example of multiple virus transmission where we see a similar phenomenon, although we don't have the extent of follow-up. So again, at baseline, no reactivity, nothing until week 24, when this gold virus here starts to be picked up. And then by 42 weeks or near a year, we have a substantial increase in the potency against both, and we start to pick up both of these viruses in neutralization. Okay, so to summarize this part of the talk, acute ART initiation, fewer than 60 days from transmission, prevents the formation of autologous neutralizing antibodies. And again, the sequences kind of predict what is going to happen. So if we have no evidence of virus escape in our sequences, we do not see subsequent development of neutralizing antibodies. However, if we have a little viral rebound, or maybe immunization in the future world, that's enough to elicit these responses, suggesting that they're primed and ready to be expanded. Early ART initiation, after about 60 days, we see that there is the development of this first ANAB response, and that continues to increase in breadth and potency over time. So it broadens from targeting that first transmitted founder virus that we've never sampled, to that first variant present at the time that we sample. And then in multiple virus transmission, we can see additional variants being targeted over time. This increases in potency of over years of antiretroviral therapy. It's not a short-lived process, but it does suggest this interesting threshold effect, suggesting that it's not just how much virus you have. Again, these acute ART initiators have virus circulating in the plasma for more than a year, in some cases, but it's this replicating virus that is required for that neutralizing antibody response to develop and then to continue on antiretrovirals. And again, we didn't go into this data very much, but HIV-specific binding antibodies, the presence of those are not enough to trigger neutralizing antibodies after ART has been started. And I think there's a lot of different mechanisms that could be proposed for this. And we're going to try to elucidate some of the reasons why ongoing virus, but is not enough to push this forward in our acute ART initiators. I'm just going to go through this real quick. I also think that this provides optimism for the fact that we could use an autologous neutralizing antibody targeting vaccine. So this is my pie in the sky. Please bear with me while I think about these future directions. So in people with HIV who have acute and early ART initiation, again, we know that the reservoirs are small and less diverse, and they have preserved immunity. And in theory, now they have this boostable autologous neutralizing antibody response. And so in theory, if we could use a vaccination that aims to boost that already primed response, rather than target a rare B cell lineage, which is kind of what our current antibody-based vaccines are doing, we may have a much more reasonable target. I mean, because we only need to be able to cover the diversity of this limited diversity and this limited reservoir, it may be an easier thing to accomplish. In addition, the vaccine world is just exploding with new discoveries. So we have, for the first time, the ability to make stabilized trimers of basically all the different viruses we want. And so for the first time, we can personalize these immunogens based on a range of envelopes that are sampled. We also know that mRNA lipid nanoparticles are extremely immunogenic, and we can rapidly turn our sequences into a vaccine product as opposed to waiting to develop all these different proteins. So my vision is that we could try a personalized autologous neutralizing antibody boosting vaccine in each of these acute or early ART initiated individuals, aiming to boost the responses against that narrow early swarm that we characterize when we diagnose and start them on antiretrovirals. So in the next 3 minutes, I'm going to tell you what I think the next study should be, because my lab is very interested in going back and forth between humans and non-human primates. And I think it's very important to see that the non-human primate model has the same sort of virus-host interactions in this regard, and therefore we could test this in a non-human primate system, which I think would be a nice intermediate step before a human trial. So this is a study looking at the effect of bNAb monotherapy in a SHIV model of persistence. So what I'm showing here is the early viral kinetics. We took 18 animals, infected them with a clade D transmitted founder SHIV. We see nice acute virus levels. They had virus replication for the first 4 months. Here we see set point levels between about 1,000 and a few 100,000. So that fairly well recapitulates what we see in HIV. We suppressed them on ART for 6 months. We either gave them a CD4 binding site monoclonal antibody or nothing. We watched these viruses return. They all rebounded. We saw a nice delay in time to rebound. This recapitulates what we see in our human trials of monoclonal antibody, of bNAb administration at treatment interruption. We suppress these animals again and then watch them rebound again, etc. But what I want to talk about is the autologous antibody responses in these SHIV-infected rhesus macaques. So at 4 months, at the time they started antiretrovirals, which is kind of similar to this cohort we just described, we looked at the plasma antibodies and the IgG extracted from the plasma and asked, does this neutralize the transmitted founder virus, the virus that establishes infection? And what we saw was, first of all, that our plasma and our plasma IgG were nicely correlated. So we could go back and forth and look at our plasma after our initiation, where we can't just look at our frank, we can look at our IgG, not our frank plasma, which has antiretrovirals in it. And we saw 10 of 18, or 56% of these rhesus macaques developed neutralizing antibodies at day 120. So this may be a little bit later than what we saw in our humans, but it's fairly similar. And so because those trends were similar, that was encouraging. So we can also look at the change in these antibodies over time. So this is our neutralization curve, kind of in a Roy G. Biv red, green, yellow rainbow phenomenon over time. And here we're plotting those antibody titers. So this is the IC50 with more potency being high and less potency being low. Over, this is our baseline time point at month 4. Over our 6 months of antiretrovirals, we see they start to increase just a little bit at the end. Then we perform treatment interruption. Before viremia, we see these tick up a little bit. And then after rebound, we see a boost, just like we saw in our human cohort. So if we look at this whole cohort of 18 animals, at baseline, we have a little more than half of these individuals with detectable neutralization. Our rebound OMS, all those viruses that broke through, whether they were treated with bNAbs or not, all of these viruses were resistant to the earlier neutralizing antibodies. This suggests that applies selective pressure and that viruses our earliest sensitive viruses cannot break through. After we have viral rebound, those responses are boosted against both of the rebound and the TF SHIV. So we see those antibodies have further adapted with just a little bit of viremia. All this is to say that our neutralizing antibody, our non-human primate is a good model to test what we think an early neutralizing antibody boosting vaccine could look like. So this is my proposed study. And I haven't started this yet, and I hope we can do it soon. But the basic concept is that we could start with our best SHIV, give a high dose infection. We can have a small amount of viremia, let's say 4 months, so that part of our animals will have neutralizing antibodies, a part of the cohort will not. We can start them on antiretrovirals, suppress their viremia. We can give vaccination after they've had a little bit of time to return to health on antiretrovirals with an mRNA lipid nanoparticle that confers an OMV that is identical to their transmitted founder virus. We can give multiple doses of this after they've been suppressed on antiretrovirals. We can then monitor for the changes in those antibody responses, see if we develop any more breadth against the swarm of viruses present at 4 months. We can look for potency of that response. We can look for cellular CD8 T cell responses. Then we can interrupt therapy and ask if we do in fact have rebound or if our boosted autologous antibody responses in early ART would be able to suppress. So we can do this in many different ways, including looking at very closely related OMs or slightly more divergent to ask how specific does our antibody or does our immunogen approach need to be. But I think this would be a nice framework to test the concept of, can we use our new vaccine technology and our understanding of autologous antibody responses to develop an autologous neutralizing antibody vaccine approach based on these data. If we could develop this, we could figure out correlates. It would potentially support the development of a human clinical trial. All right, that's all. Thank you so much for your attention. I want to thank the people from my lab, Ryan Krause and Greg Whitehill, who did the majority of this work. Rebecca was essential in this work. And then our partners at UCSF who provided the samples and a lot of the clinical information. With that, I'll say thank you, and I'm happy to take any questions.
Wow, thank you so much, Dr. Bar. That was quite an amazing presentation. I'm still trying to process it all, but I'm gonna open it up to people more intelligent than me to ask the first question. If anybody has a question for Dr. Bar, just unmute yourself and please fire away.
Hey, Katie, that was such a nice talk. I don't know if you can hear me, but I was wondering. Hey, Katie. I was wondering if you could talk a little bit more about the personalization at the end, because I kind of hadn't seen you present that before. And I was just wondering if you think there is a route where there would be sort of like a one product that would stimulate autologous antibodies, or by definition, they're targeting epitopes that are just inherently not conserved and it has to be personalized in the sense that each person's virus's vaccine would be different.
Yeah, I think that would be the goal of the non-human primate study, would be to test just that. So we would use multiple shivs. So we'd have distinct viruses. And then for each one of those shivs, you could have a personalized matched to either the early or the transmitted founder virus, and then another that's just maybe a within clade. So for our clade C virus, we'd have sort of a consensus clade C immunogen. And you could test whether you needed a very close match or whether that initial response will be boosted even with a more divergent immunogen. And I think that would be a key question for how you could move forward. I do think personalization for the first time is actually possible, because we really have, as a vaccine field, an HIV vaccine field, for the first time developed the ability to make stabilized OMV trimers to a range of viruses. I mean, we feel like we've had SOSIPs for a long time, but really we had BG505 for a while. And then we started to have a few more viruses that would hold, with the mutations we knew would hold their conformation. But now structural biology has really advanced so that we can take almost all OMV sequences and make an OMV immunogen. And so hopefully that technology continues to evolve. But we could try a personalized OMV as well as just any sort of clade matched or relatively close OMV, and ask if we can get the same boost to the autologous response. And I think the non-human primate model is the right model to test that, because we can look at multiple viruses. We'd know the virus, we'd know how divergent the boost would be. But translating that to a human trial, you know, if we have all of these participants for this 23-person study were diagnosed at acute or ART initiation and started on our ART on that day, you could take that plasma sample from that day, start the person on ART, let them have 6 months of virus suppression on ART, return to health. And then at that point, ask, would you like to continue with this process? Because over that 6 months, you could develop that immunogen, make sure it was stable, make sure it was ready to go, and deliver that via mRNA lipid nanoparticles after they've been stabilized for 6 months. So that's the theoretical way that you would deliver it with humans.
Very cool. Thank you. Great talk.
Thanks, Rebecca.
Would these autologous neutralizing antibodies be targeting just one epitope, or would they be targeting like the V2, V3, and CD4 binding domain?
That's a good question. So we think the first strain-specific autologous neutralizing antibodies tend not to target these BNAb epitopes that we have well described, V2, V3, CD4 binding, etc. They tend to target much more narrow, sort of highly mutable regions of variable loops, etc. But I think you can see from that multiple virus transmission participant, eventually on ART, the response broadened to target divergent viruses, meaning that either there's one response that's able to handle relative breadth, or there's multiple responses that equally well target distinct viruses at different epitopes. So we don't know, we haven't mapped the actual epitopes that they target. That's one of the things we're working on with some of those participants. But I would not expect they're actually targeting broadly neutralizing epitopes.
Okay. Right.
Yeah, go ahead, Alan.
Hi, Katie. Thank you so much for coming back. I remember when we invited you years ago, and we had dinner with Rebecca, and it was such a nice evening. And thank you for your presentation tonight. It was actually an opportunity for people who don't do this every day to understand the complexity and importance of the work you do. My question is just an opportunistic one. I've been hearing about your collaboration with Rebecca and for years. Now that I sort of understand what you were saying, how does the work you're doing with Rebecca relate to the work that you presented this evening?
Yeah, so Rebecca and I have a UO1 together. We have the other grants together as well, but the larger one we're working on is trying to understand how we can improve broadly neutralizing antibodies by making them less susceptible to resistance evolving. And so for the clinicians in the room, if you remember back in early antiretrovirals, when we would have sort of opposing resistance patterns with thymidine analogs, you know, with AZT and with some of our other antiretrovirals, and we thought about that mutual phenotypic antagonism where we combine like a 3TC and an AZT together because the virus can't escape from both of these at the same time. So trying to harness that concept of, you know, escape pathways that are mutually exclusive within BNAb world, as opposed to with our small molecules. So what we're trying to do in this very systematic multi-step approach is to identify broadly neutralizing antibodies that are complementary, because a virus cannot escape from both. So whether these are related, they have, you know, they're targeting a similar but slightly different epitope, maybe at a different angle, maybe they have, you know, for instance, there are CD4 binding site antibodies, but different classes that have slightly overlapping epitopes such that you can change one and sensitize to the other. Anyway, we're trying to develop those BNAbs to be better, the combination of BNAbs. But we're also trying to figure out the role of autologous neutralizing antibodies and how they may help or hinder BNAbs. And so part of that is a broad-ranging study of autologous neutralizing antibodies. And access to this acute cohort gave us, it's just a little bit of a cleaner system to understand antibodies early on before it gets too muddy. And so that was a good understanding, a good way for us to think about, well, all of our participants are going to be on antiretrovirals, and what happens to that antibody response with antiretrovirals, because it's surprisingly not well understood how antiretrovirals affect some of our immune processes that are very much antigen dependent. That was a long answer.
Oh, that was fascinating. Thank you. I actually followed that. I appreciate it. But I'm going to leave the other questions to any basic scientists who are on the phone.
Does anybody else have any other questions? I'm going to ask a really simple question that I think is going to document my ignorance. Is that in the acute ART treated cohort, is there any downside to preventing the development of autologous broadly neutralizing antibodies or neutralizing antibodies? Is there? No.
I don't think so. I mean, I think we should treat every person who is diagnosed with HIV as soon as we possibly can. There's overwhelming evidence that that's beneficial for them, that's beneficial for public health. And I guess the encouraging part here is if you don't have, if you start antiretrovirals early enough that you don't develop neutralizing antibodies, we think with just a little bit of a push, you can develop those. So that response has started to be primed. And so hopefully immunization could build on that in a way that wouldn't be harmful. We also know that acute ART initiation preserves immunity, very small restricted reservoirs. So, you know, there's this question about, is there a sweet spot where you've developed HIV-specific immune responses, CD8 T cells, and autologous antibodies, but also had very early ART so that you preserved all of that immunity? I mean, I think that's important for vaccination and immunotherapy and whatnot, but for just general individual and public health, we should start antiretrovirals as early as possible. And for sure that outweighs any theoretical potential for post-treatment control or good candidacy for some theoretical thing that's going to come down the line.
Yeah. Well, thank you. If no one else has any questions, I don't want to keep people longer than necessary, but
Mark,
again, I really, yes, David, go ahead.
I do. Thanks so much. That was fascinating. I'm still, like Mark, trying to process it. So for this strategy to work, the vaccination strategy to work, I assume that you would have to administer the vaccines in the background of complete viral suppression, right? Because otherwise wouldn't, if you're giving the trimer based on the founder virus, the infecting virus, then when by the time you actually develop the antibodies, if there is replicating virus at the time, it would just have escaped by the time. Yeah. So you would,
that would be my goal.
Have to be done on the background of complete viral suppression.
Yeah, I think we'd need virus suppression, but I think the vast majority of people who start antiretrovirals and are able to take them have complete virus suppression. Even folks where we see little blips, that's probably not replicating virus. That's probably just virus stochastically being expressed from cells, but not leading to multiple rounds of infection. Can you hold on one sec, sorry? So I don't think that that's, I think that is a bar for many people. That is not everybody is able to maintain virus suppression through antiretrovirals. But I think the vast majority of people who can adhere and who would want to participate in a clinical trial like this, and ultimately to try a vaccination, would be able to sort of participate in that level. And nowadays, you know, we're getting long-acting injectables, we're getting longer-acting medicines, so adherence is even sort of maybe easier or multimodal, and hopefully we'd be able to sort of guarantee virus replication is suppressed if we were to immunize to that original virus and hope it doesn't continue to evolve.
Thank you.
All right, anybody else? Okay, well, again, I want to thank Dr. Bar. Thank you for taking the time out of your busy schedule. We really appreciate it. It was really informative, and I think we'll all be mulling over what you showed us for a while, trying to understand, you know, and grasp its potential for the future. So very exciting stuff. Appreciate it.
Thank you.
All right, have a good evening, everybody. Thanks for joining. Take care.